So today, you guys, I want to review our Clover experiment to just kind of set you up to review for the test that you have tomorrow. Initially, the test was supposed to be today, but when a couple of things happened, and I just realized that it wasn't really a best idea to give you guys the test after a 4-day break. So tomorrow, you're going to take the test on procedures and variables and hypothesis and testable question. So those things are all together tomorrow on a quiz. You're going to actually take it in 3 different parts, but we'll talk about that tomorrow. So today we're just going to review our Clover experiment to review the different parts of a science experiment so that you can see again how we do this. So we actually have data, so we're going to talk about the data today. So first of all, question. In our experiment, our question was, does the amount of water affect the time it takes clover to germinate? So one of the things we need to talk about is how do we know that this is a testable question? So how we know that this is a testable question is because it has something that we can collect data on and it has something that we have changed. So the thing that we can collect data on is the number of days it takes clover to germinate, and the thing that we changed is the amount of water. So when we talk about then, the next thing we needed to do after we identified our question was we need to identify our variables. So we need to identify the independent variable, which is the thing that we are going to change. We need to identify the dependent variable, which is the thing that we're going to measure. We also talked about controlled variables, and the controlled variables are the things that we're going to keep the same. So all the things that will keep the same. And I'll talk about those as we go through today also. So our independent variable here is going to be the amount of water. And the dependent variable is the time it takes the clover to germinate. So the independent variable, the thing that we're changing, is we're going to change the amount of water. And the dependent variable, the thing that we're going to measure, is the time or the number of days it takes the clover to germinate. So the next thing after that is we wrote a hypothesis. Now the null hypothesis and alternative hypothesis is the new way that we do this. And if you remember, you have to do this by knowing your variables first. And when we write our independent or the null hypothesis, we just basically put our independent and dependent variable into a formula. So the formula is written right here, and it says right here that the independent variable has no effect on the dependent variable. So what we have to do is take our independent variable, the amount of water, and just insert it into this space. And then we have to take our dependent variable, the time it takes the clover to germinate, into this space. So our null hypothesis then would be the amount of water, our independent variable, has no effect on the number of days until the clover germinates, which is our dependent variable. So it's just this formula, and I inserted the independent and the dependent variable. So that's how we get to our null. And the reason we need a null hypothesis, if you remember, is so that we can say we understand that there may be no connection between those two things. There may not be any reason to study this. Okay. The alternative hypothesis then is to say these are the other possible things that can happen in our experiment, and we have to determine how many. So remember that to write a hypothesis, we need to know again what our variables are, and then we need to know the many different versions of the independent variable, or how many different versions of that independent variable we're going to have. So that tells us how many hypotheses we need to write, and then we need to know what are we going to do with the dependent variable. So to determine the amounts, how many alternative hypotheses we need to know how many different parts of the independent variable. So for our experiment, we watered our clovers with 5 milliliters and 10 milliliters and 20 milliliters of water. So that tells us, because there's 3 different variables here, 1, 2, 3, that we're going to need to write 3 different alternative hypotheses, one for each of the different amounts of water. So to do that, we already talked about that, we need 3. We're going to take those and we're going to write them down. So we're going to say that plants watered with 5 milliliters, and then we're going to say the same thing after all of them. We are going to say will germinate in the fewest days, or we could say in the most days, but this one seems to make the most sense. So all three hypotheses, one for the 5 milliliters, one for the 10 milliliters, and one for the 20 milliliters, and then all the rest of it stays the same. So plants watered with 5 milliliters will germinate in the fewest days. Plants watered with 10 milliliters will germinate in the fewest days. And then our H3 is plants watered with 20 milliliters will germinate in the fewest days. So if we were to look at all of our hypotheses, in order to be correct, we need a null, and then an H1, an H2, and an H3, and you can see them there. So our null hypothesis, using that formula, the amount of water has no effect on the number of days until the clover germinates. Our H1 is plants watered with 5 milliliters will germinate in the fewest days. Our H2 is plants watered with 10 milliliters will germinate in the fewest days. And then our H3 is plants watered with 20 milliliters will germinate in the fewest days. So if you notice, the next step after hypothesis then is procedures. And when we write our procedures, now that we have identified our variables and we've written our hypothesis, now we need to write our procedures. And if you remember, the 4 things we talked about is that they need to be short and sweet directions. Detailed, very short, very easy to read, nice and simple. Second, you need to number your steps. So you need a 1, a 2, a 3, a 4, however many you need. You want to start with an action verb or a word or a verb or an action word, and then you want to make sure that you include details and measurements. So you guys watched this in a video, but these are our actual procedures. Label the pots that say 5 milliliters. Place labeled pots that say 5 milliliters of water. Place labels on 5 different pots that say 10 milliliters of water, and then do the same for the 20 milliliters of water. We placed a paper towel at the bottom of each pot. Number 5, fill the pots with 6.5 centimeters deep with topsoil. Should be to the ridge in the pot. Step 6, sprinkle 1 milliliter of seeds onto the center of the soil. Step 7, add another 2 centimeters of soil to the pot. That should be up to the second ridge, and then repeat steps 5 through 7 for the remaining 14 pots. Step 9, set the pots in the same location under the grow lights. Step 10, program the timer on the grow lights to run 12 hours a day. Step 11, water the pots every day by pouring the amount of water on the label of each pot into the center of the pot using a graduated cylinder. And then number 12, record when the clover has sprouted through the surface of the soil. So over the weekend, I took these pictures. So these were it's hard to see that, sorry. Day 1, day 2, day 3, day 4, day 5, and day 6. And you can see that they didn't do exactly as we'd hoped. So this was the data that I collected. And so you can see that the 5 milliliters, pot 1 took 5 days to germinate, pot 2 took 5 days, pot 3 took 6 days, pot 4 took 6 days, and pot 5 took 6 days, and that's with 5 milliliters of water. With 10 milliliters of water, all 5 pots germinated on day 4. And then with the 20 milliliters of water, most of them were day 5 and 1 was day 4. So what we would want to do to look at our data then is to calculate the average. And if you don't remember how to do average, it's the same word as the mean. So the average or mean, you add all the numbers up. So for 5 milliliters, I would add up 5 plus 5 plus 6 plus 6 plus 6, which gives me 28. And then I would divide that by the number of pots that I have, and that was 5. So when I divide that out, I'll get my average. So I did that for all 3, and here's our averages. So the 5 milliliter pots took an average of 5.6 days to germinate. The 10 milliliter pots took an average of 4 days to germinate, and the 20 milliliter pots took an average of 4.8 days to germinate. So now the next thing we need to think about is, well, which hypothesis then is supported by our data? So which one germinated in the fewest days? Now, if they were all exactly the same, we would accept our null hypothesis, because that's saying that the amount of water has no effect, saying that all of them would germinate at the same time. And that wasn't the case. So then we look at our H1, our H2, and our H3. If we go back, our 10 milliliters of water, we can see it right here, 10 milliliters of water germinated in the fewest days. So we want to go here, and we can see then that we would accept our H2 in this situation. So our H2 is the hypothesis that our data supports, that The ones that were watered with 10 liters will germinate in the fewest days. So if you want to go back, we can talk about that for tomorrow's video or tomorrow's quiz, you need to understand what a testable question is, which means that a testable question means that you can collect data and you change something. So it has an independent and a dependent variable. For variables, you need to understand the independent variable and the dependent variable. So the independent variable is the thing you change. The dependent variable is the thing that you measure. The controlled variables are all the things that you're going to keep the same. And that's one thing that I didn't talk about. So in our experiment, we kept the same the amount of soil, the type of water, the type of pots, the amount of light that they were going to get, when we watered them, how often we watered them. All of those things were kept the same, so those would be considered controlled variables. So those are the 3 types of variables. Then we talked about hypotheses, and we have a null hypothesis, which states that one has no effect on the other. And then you have the alternative hypotheses, and the alternative hypotheses are determined by how many different versions of the independent variable you have. And in our case, we needed to write 3 alternative hypotheses. So you need to be able to identify those, and that's your prediction. And then lastly, procedures. The 4 things you need to remember is they need to be short and sweet directions. You need to number your steps. You need to start with a verb or an action word and include details and measurements. And then lastly, we just talked about data collection, and that part is not on the quiz tomorrow. So that's just through procedure. So again, you can use this to review, and then you're also going to look at Quizlet today to review. I hope that helps you to understand again and review before the quiz tomorrow.